boys and girls. I'm Grandma Bertha. What? Oh, well, what's that? What am I reading? Oh, I'm reading this here biology book. My doctor told me that I need to read it because I don't know nothing about what's going on in my body, so I'm teaching myself. Let's see. I'm reading about what's it called? Oh, sensory and motor mechanisms. I'm getting educated on this. Let's see. Let me teach you too. You could help me for my doctor's appointment. It's on. What's the day? What's today? Thursday? Maybe it's next Wednesday. Oh, heck, I don't know. Well, I'm first going to tell you about sensory pathways.
Now, let's see here. Oh, okay. This book says that sensory pathways are essential in detecting signals and sensory information and outputting the correct physiological movements in response to the correct signal. This provides the foundation physiologically for all animal behaviors, and my behaviors too. Sensory pathways include 4 functions: reception, transduction, transmission, and perceptions. Each sensory process begins with a stimulus. Stimuli are forms of energy. They are first obtained by sensory receptors, which are sensory cells that interact directly with the stimulus. They are responsible for converting stimulus energy from outside and inside of the body into signals that are transmitted to the central nervous system, or the CNS, where a motor response may be generated.
My doctor says that sensory receptors aren't working as brightly as they used to. What a nincompoop. When I was your age, all young and spry, I would have given him what I call a knuckle sandwich. For talking to me like that, that's just rude. Well, but anyways.
Animals have many sensory receptors that are used to detect a countless variety of stimuli. Every kind of stimulus, no matter what, results in an opening or closing of ion channels. When a stimuli or substance binds to a chemical receptor outside of the plasma membrane, a flow of ion drifts across the membrane, changing the membrane potential, also known as receptor potential. The membrane receptor potential have varying magnitudes based on the stimulus's strength. The conversion of a physical and chemical stimulus to a change in membrane potential is called sensory transduction. When a sensory receptors transduce or convert stimulus's energy into receptor potential, they initiate transmission of action potentials to the CNS. Action potentials are triggers produced by neurons, which act as sensory receptors and have axons, or extensions, that connect to the CNS. Action potentials produced by afferent neurons, or sensory neurons, are formed from non-neuronal sensory receptor cells that form chemical synapses which respond to stimuli by increasing the action potential production rate. And the last course the stimulus has to take when the action potentials are reached the brain. They are brought to the brain by sensory neurons, powered by circuits of neurons, which generate a stimulus's perception. Perceptions are constructions formulated in the brain, like colors, sounds, smells, and tastes, and don't exist outside of it. This is possible because action potentials form sensory receptors, and they travel along particular neurons that belong to certain stimuli. These neurons synapse with other particular neurons in the brain and spinal cord. From there, the brain distinguishes stimuli, sight, sound, smell, and taste, solely via the paths each action potential has traveled.
I have a good example of this. It was just yesterday. My husband Earl was a hootin' and a hollering at me while I was sewing.
Hey, yo, Bertha, you're burning something.
Huh? Oh, dang nabbit. Oh.
the stimulus of the burning food went up my nose, which is the stimulus, and perception, when the stimulus reached my brain, helped me see the burning food in my head. So I dropped my quilt real quick and sprinted to stop the food from burning. That's the difference between perception and reception. The difference is that reception is when the stimulus is detected by sensory cells, and perception is when that stimulus is distinguished by the brain.
My doctor told me that my sense receptors in my ears aren't working as well as they used to. But I think Earl's receptors are worse. I'll tell him, "Hey Earl, what do you want for breakfast?" "Earl! Hey Earl!" Oh, what a nincompoop. "Hey Earl, what do you want for breakfast?" That's when I told Earl that he needs a hearing aid because his ears aren't working as properly anymore. He told me I didn't know anything about his ears or anyone's ears, so I looked it up.
In mammals, there are several components that make up the structure of the ear. Without these structures, mammals would not be able to hear at all. There are 3 parts to the ear: the outer ear, the middle ear, and the inner ear. The outer ear contains the ear canal and the tympanic membrane, which vibrates when moving air rushes across it. Some mammals also have a pinna, which is an external flap and collects sounds. The middle ear has 3 bones: the auditory ossicles, the malleus, the incus, and the stapes, also referred to as the hammer, the anvil, and the stirrup. These 3 small bones send the vibrations to the oval window of the ear, which is a membrane on the cochlear surface. The middle ear consists of the eustachian tube, which links the outer ear to the middle ear. The inner ear has 2 parts: the cochlea, where the hair cells vibrate in response to fluid movement in the cochlea, and the vestibular organ, which gives animals their sense of balance. When the stapes vibrates against the oval window, it creates pressure waves in the fluid of the cochlea. The waves push against the cochlear duct and the basilar membrane. The B membrane, which is so called, and attached hair cells vibrate up and down. With each vibration, mechanoreceptors in the hair cells open and close ion channels. The ion channels travel to the auditory nerve, which connects to the brain. Hair cells bending in one direction increase neurotransmitter release and action potential frequency to the auditory nerve. Hair cells bending in the other direction decreases both, and therefore decreasing auditory nerve sensations.
Then I read on to learn about other animals and their ears. I also found out that the function of the ear doesn't just help with hearing, but for balance. Maybe that's why I need this doohickey here. Every time I try to walk, I stagger and fall. Oh dear, I gotta go to the bathroom. Hey girl, can you get my walker for me? It's all the way over there. Earl. Oh, this nincompoop. Help! Oh, Earl!
help?
Now, why I need these glasses here is because I can't see too well. So my doctor told me to look this up too. So let me tell you about the structure of the eye. First, we're going to look at invertebrates. Yes, that's what it is. They all have light-detecting organs, or some sort of light-detecting organ. They're called ocelli, which are eye spots that only receive light through the opening where there aren't any pigmented cells, which block the light. They also have compound eyes, most commonly in insects and crustaceans. And let's see, they say here that the compound eyes have thousands of light detectors called omnatidia, which have light-focusing lens. They're extremely effective at detecting movement, which insects need to get away from fly swatters and such. And also, invertebrates have single lens eyes. They work like a camera, so they contain a pupil where the light enters, and an iris which contracts and expands to control how much light the pupil lets in. And the muscles in invertebrates' single lens move forward and backwards to focus. Now let me tell you about vertebrates. And all vertebrates have a single lens eyes. Their eye structure is, we're going to go based off the human model according to this here book. It says the eye is surrounded by the conjunctiva, which is a mucous membrane, and the sclera, which is a connective tissue and makes up the whites of your eye. The choroid has a thin pigmented layer, and so in the front of the eye, the sclera forms the cornea and the choroid forms the iris, which, as I said before, regulates how much light enters the pupil. And the pupil is the hole in the middle of the iris. Inside the choroid is the retina, which contains neurons and photoreceptors. And the photoreceptors are rods and cones. And light, or photons, strikes the retina, passes through the neuron layer, and reaches the rods and cones. And neurons relay the visual information from photoreceptors to the optic nerve and brain. The choroid and the retina forms the innermost layer of the eye. And close to the retina is the fovea, which helps provide the sharpest vision. Well, mine certainly aren't working that well. And contains cones and is the center of the visual field. And the optic nerve exits at the optic disc and leads to the brain's primary visual cortex. And last is the lens, which is a transparent disc of protein which divides the eye into two cavities, which is the aqueous humor and the vitreous humor. Now let me tell you about the skeleton types, which provide help in our motor mechanisms.
Skeletons in animals are vital for movement, support, and protection. They maintain an organism's shape and protect the soft tissues like the brain and the heart. There are hydrostatic skeletons that contain fluid that's held under pressure in a closed body compartment. It's found in cnidarians, flatworms, nematodes, and annelids. They use their form to control movement by use of their muscles, which change the shape of the fluid-filled compartments. They are best suited for aquatic environments, but they can provide crawling and burrowing support on land as well. Then there are exoskeletons, which are hard encasements that are exposed to the animal's surface. Bivalves like clams, insects, and some arthropods, which are crustaceans, spiders, and other insects, have exoskeletons. Insects and arthropods have a cuticle, which is a joined exoskeleton secreted by the epidermis, and bivalve shells are made of calcium carbonate secreted by the mantle, which is a sheet-like extension of the body's wall. The cuticle is made of chitin, and it's mixed with a protein matrix, which allows the composite material to be created. And exoskeletons are molted or shed whenever the arthropod grows. And last, there are endoskeletons, which you and me have. They are hardened skeletons that are internal, rather external, that's buried within their soft tissues, in animals from sponges, echinoderms, and chordates to animals and other mammals have them.
Now let me tell you about muscle contractions. Let's use the sliding filament model, which can be used to explain the whole muscle contraction by elaborating on the contraction of one sarcomere. Sarcomeres are repeating units of dark and light bands in skeletal or striated muscle. The thick and thin filaments, the thick containing myosin and the thin containing actin, slide past each other and increase their overlap. This sliding relies on the myosin and actin interaction. Myosin binds to form actin and makes a cross bridge, pulling the thin filament towards the center of the sarcomere. The bridge is broken when a new ATP binds to the myosin head, cleaves to it, then binds again to actin. This cycle of binding and release needs to be repeated in order to get muscle contraction. Calcium ions and actin-bound proteins play critical roles in muscle contraction and relaxation. When the calcium concentration rises in the cytosol, the thick and thin filaments slide past each other and the muscle contracts. But when the concentration falls, binding sites are covered and the muscle relaxes. Now, the last thing I want to talk to my doctor about is the fast versus slow muscle fibers, because I think that I have too many slow fibers now. So let's see. Oh, here it is. This talks about the fast twitch and the slow twitch fibers, and there are variations in the contraction of each. Fast twitch fibers develop tension 2 to 3 times faster than the slow twitch. They enable brief and rapid and powerful contractions. They can be glycolytic or oxidative. And here's an example. For example, if a muscle is used repetitively for something, some fast glycolytic fibers can change into fast oxidative fibers to keep muscle from getting fatigued too quickly. And slow twitch fibers are found in muscles that maintain posture and can sustain longer contractions. It has less sarcoplasmic reticulum, but pumps calcium more slowly than fast fibers. Because the calcium, or Ca2+, stays in cytosol longer, slow twitch muscle fibers can last 5 times longer as in one fast fiber. Well, that's it, boys and girls. Thank you for listening to me. I know exactly what to say to my doctor now on whatever day I'm going. Well, I have to use the bathroom now. You kids best be on your way, but first give Grandma Bertha a hug. Thank you, sweet peas. All right, well, bye now.